A very warm welcome to all the students. With this session, we are going to begin our discussion on the classification of the animal kingdom. We are going to discuss a general account of the classification, various bases on which the animal kingdom has been classified, the details of phylum Protozoa with some of its important examples. In the classification of the animals, we group together similar animals on the basis of their properties. The members of each group have some structural features in common. Actually, the practice and science of classification is known as taxonomy. This classification of the animal kingdom is a morphological classification, in which similar animals are grouped together on the basis of their structural properties. So first of all, let's get acquainted with the various taxonomical ranks that we are going to use in the classification of the animals. The smallest group, which is formed on the basis of similarity in structural properties of the animals, constitutes a morphological species. Species with similar characteristics form a genus. Similarly associated genera constitute a family. Closely related families form an order. A group of similar orders forms a class. A group of classes is known as phylum. A group of phyla constitutes a subkingdom, and the subkingdoms together constitute the animal kingdom. The animal kingdom has been divided into two subkingdoms. The first one is subkingdom Protozoa, and the other is subkingdom Metazoa. Protozoa includes unicellular, eukaryotic animals with a single phylum, Protozoa. Its examples are Euglena, amoeba, paramecium, etc. We are going to discuss the details of phylum Protozoa in this session. The other subkingdom is Metazoa. It includes multicellular animals. On the basis of level of organization, Metazoa is further divided into two branches. The first one is branch Parazoa and the second is Eumetazoa. In Parazoa, the cells are loosely aggregated and do not form true tissues or organs. It includes the phylum Porifera, whose examples are sponges. The second branch is Eumetazoa, which includes truly multicellular organisms with organ and organ system level of organization. It includes phyla Coelenterata to Chordata. Based on symmetry, Eumetazoa is further divided into two groups, Radiata and Bilateria. As the names indicate, Radiata includes those animals which are radially symmetrical. They include phyla Coelenterata and Ctenophora. Whereas the group Bilateria includes those animals which are bilaterally symmetrical. They include phylum Platyhelminthes to phylum Chordata. Now let's see what exactly is radial symmetry and bilateral symmetry. In radial symmetry, the animal can be divided into two identical halves by any of the radial planes along anterior-posterior axis of the body. Whereas in bilateral symmetry, the animal can be divided into two identical halves only by a single plane, passing from dorsal side to ventral side and along the anterior-posterior axis of the body. So the animal can be divided only into two identical left and right parts. Animal kingdom has also been classified on the basis of germ layers. Germ layer, which is also known as germinal epithelium is a group of cells formed during the animal embryogenesis. So on the basis of germness, animal kingdom has been divided into two types. One is diploblastic and the second is triploblastic. Diploblastic animals have two germ layers, the outer ectoderm and the inner endoderm, with a non-cellular layer known as mesoglia between the two. Diploblastic animals consist of phyla Coelenterata. The other group is triploblastic. As the name indicates, it consists of three germ layers, the outer ectoderm, middle mesoderm, and the inner endoderm. This group consists of animals from phylum Platyhelminthes to phylum Chordata. The animals included in Bilateria have been further classified on the basis of the body cavity or coelom. A true coelom is a body cavity lying between the gut and the body wall and is lined with coelomic epithelium. Now, depending upon the presence or absence of a true coelom, Bilateria has been divided into three categories. First is acoelomata, second is pseudocoelomata, and the third is eucoelomata. Acoelomata consists of those animals in which coelom is absent. Its example is phylum Platyhelminthes. In pseudocoelomata, the animals consist of a false coelom, that is a body cavity which is not lined with coelomic epithelium. Its example is phylum Aschelminthes or Nematoda. The third category is eucoelomata, which consists of a true coelom, that is a body cavity lying between the gut and the body wall and lined with coelomic epithelium. Now the coelom in eucoelomata is of two types. First is schizocoelomata group, which consists of those animals in which coelom develops by splitting up of mesoderm. It consists of phyla Annelida, Arthropoda, and mollusca. The second group is enterocoelous coelomata, in which the coelom is enterocele, which originates by fusion of pouches of the embryonic gut, that is archenteron. It consists of the rest of the phyla. If we look at the cross section of this acoelomate, we can see that there is this gut which originates from endoderm, outer ectoderm, and the middle muscle layer which originates from mesoderm, but there is no body cavity or coelom present. This is the cross section of a pseudocoelomate in which we can see a cavity which is present, but it is not a true coelom since it is not lined with coelomic epithelium. Therefore, this cavity is known as pseudocoel. This is the cross section of a coelomate in which we can see the outer ectoderm, the inner endoderm which differentiates into gut, and the middle mesoderm which differentiates into peritoneum and muscle. There is a cavity lying between the gut and the body wall, and since this cavity is lined with coelomic epithelium, it is a true coelom. The coelomates, that is, those animals which consist of true coelom, have been further divided into two types based on their development with respect to blastopore. Blastopore is an opening found in the archenteron during the embryonic period. So the first type are the protostomes. These are those animals in which the embryonic blastopore develops into mouth. The examples are the animals of the phyla Annelida, Mollusca, and Arthropoda. The second type are the deuterostomes, in which the